Hello, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Yuki Shindo, and I'm a postdoc at Dartmouth, and will be moderating today with Rashmi Patel, a postdoc at National Cancer Institute. We are excited to highlight the work of our outstanding postdoctoral members. Today, Lydia Djenoune from Massachusetts General Hospital, Harvard Medical School, and Granton Jindal from UC San Diego will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. So it's my pleasure to introduce Dr. Lydia Djenoune, who is currently an instructor in Shru Yun's lab at Massachusetts General Hospital, Harvard Medical School. Lydia did her graduate training at the French National Museum of Natural History, where she studies the molecular and morphological basis of spinal CSF contacting neurons. After her PhD, Lydia moved to Boston to begin her postdoctoral training, where she first focused on understanding the role of calcium signaling in kidney morphogenesis, while she is now focusing on understanding the role of cilia and mechanosensing mechanisms that instruct left-right asymmetry. She's received several fellowships and awards, including a postdoctoral fellowship from the American Heart Association and a Career Development Award from the American Heart Association. We really look forward to your talk, Lydia. Please begin when you're ready.
So hello, everybody. Thank you, Yuki, for this kind introduction. Before starting my talk, I would like to thank the SDB Ethel Browne Harvey Postdoctoral Seminar Series organizers for the wonderful opportunity to be here with you today to share with you my work that will be centered around investigating ciliary mechanosensation in left-right asymmetry. But first, I would like to tell you why I'm interested in left-right asymmetry. Apologies, yeah. So I am interested in left-right asymmetry because despite an apparent external symmetry, vertebrates exhibit a striking left-right asymmetry in the structure and in the position of their internal organs, with, for instance, the, oops, just give me 1 second. Here, you should see it, yeah. Oh, I'm sorry, it's a bit lagging, but okay. Despite an apparent external symmetry, vertebrates exhibit a striking left-right asymmetry in the structure and in the position of their organs, with, for instance, the heart and the stomach on the left and the liver on the right. And defects of left-right asymmetry fall in the spectrum of a syndrome called heterotaxy, which is defined by the abnormal arrangement of internal thoraco-abdominal organs across the left-right axis of the body. And heterotaxy is actually not a trivial organ, as its prevalence is reportedly in 1 to 10,000 births, and it's typically associated with cardiovascular malformation. And to illustrate further the importance of proper lateralization, you have about 90% of patients born with heterotaxy, which are also born with the most common survivable birth defect, which is congenital heart disease. That is a disorder resulting from abnormal heart development. So now that we know that proper lateralization is important, where does it all start? How does it take place? So it all starts in a structure that is ciliated and transient and conserved across vertebrates called the left-right organizer. This organizer is also called the Kupffer vesicle in zebrafish or the embryonic node in the mouse. And here I'm going to give you a brief overview of the main steps that take place to break the initial left-right symmetry. So the first step that is taking place is formation of cilia in the structure. Those cilia beat to generate a leftward flow in the mouse node and a counterclockwise flow in the zebrafish left-right organizer. This flow is somehow transduced on the left side of the structure. And this transduction leads to an asymmetric release of calcium that leads to an asymmetric cascade of gene expression called the nodal cascade. And this ultimately leads to proper organ cytosis. And despite extensive work that has been performed to understand each of those steps, there are still some gaps that remain, especially at the level of the flow transduction. We still don't really know how the flow is transduced into proper lateralization. And this is the main focus of my work. But first, to understand how the flow is transduced, let me go back to the first 2 steps of this model. So as I mentioned, I'm so sorry, like things are lagging a little bit. You'll have to be patient. Okay, so sorry, you'll have to bear with me. My computer refuses to cooperate, but okay, so in the structure, you have two types of cilia. Immotile cilia that are highlighted in this movie by leftward flow or counterclockwise flow. And we can visualize the leftward flow here in this video thanks to injections of fluorescent beads that we can map with the standard deviation on the images on the right. And you have to be aware of the fact that at early stages of development, the leftward organizer is mainly characterized by immotile cilia that you can see in blue in the bottom pictures. And as the structure grows and develops, those immotile cilia are slightly replaced by motile one. And at the end, the structure is mainly composed of motile cilia. So our group also reported that in addition to leftward and counterclockwise flow, using genetically encoded calcium indicators, our group reported that the earliest molecular events taking place to break the left-right asymmetry were calcium oscillation in the cilia located on the left anterior side of the left-right organizer. Those oscillations that you can map with those traces that represent the variation of fluorescence over time. And when we looked at the spatial temporal distribution of those calcium oscillation in the left-right organizer, they're mainly taking place between the 1 and 4 sonmite stage when you have mostly immotile cilia in the structure. And we also demonstrated that those oscillations were important for proper left-right asymmetry. And here I'm showing you that those oscillations of left anterior sided in the structure. And using calcium buffers, when you block those oscillations, you actually lose the proper lateralization. And in the fish, the heart is located on the left side. So if you block those oscillations, it doesn't mean that you end up with the right-sided heart. It means that the heart will be completely randomized. And in addition to those calcium, we also show that those calcium oscillations require the polycystin 2 cation channel PKD2 and that they are flow dependent, because when you use a knockdown approach to block the polycystin channel 2, or when you block ciliary motility, you actually lose the left-sidedness of those oscillations. And instead of being systematically on the left anterior side, those oscillations will be lost in the absence of polycystin 2 or randomized in the absence of ciliary motility. And this leads to a complete randomization of the heart location, truly demonstrating that those oscillations are crucial for proper lateralization. So those results lead to the following question: how are those left-sided intracellular calcium oscillations generated? And because they're located in the side of the structure that is subjected to the highest amount of flow and force, we're hypothesizing that left-right organizer cilia are mechanosensors responding to the flow and transducing it in calcium oscillation and into proper left-right asymmetry. So to test this hypothesis that cilia are mechanosensors, we needed a method to specifically bend the cilium. And because traditionally, to test mechanosensation, you have approaches where you apply artificial laminar flow, but this approach wouldn't specifically target the cilium, as it might potentially also target the cell membrane, and it might also transport chemical cues. So you wouldn't be able to decipher whether you have mechanosensation or chemosensation. So to test the hypothesis that cilia are mechanosensors, we decided to apply an optical approach, and we decided to then apply to use optical tweezers to test this hypothesis. So what are optical tweezers? They're a biophysical tool that enables precise non-micron manipulation. It's all optical, and it's purely based on the fact that light has momentum, and that changes of light momentum generate optical forces that are capable of moving objects that are micron-sized in the range of piconewtons and in distances in nanometers. So I'm really sorry. Okay, so you won't really see the movie playing. So here you should see, that a bead is actually trapped in the optical tweezer. So we turn on the laser, and this allows us to trap the bead that is in the beam of the laser. So following this approach, we decided to bend cilia. And here I'm going to show you 3 examples, 2 in cell culture and 1 in zebrafish left-right organizer in the middle. And here you should actually be aware of the fact that those cilia that you see moving are not supposed to be moving. They are only moving when we turn the laser on and when we move the laser beam in a direction that we want to, that we are actually controlling. So here I'm going to ask you to focus on the movie that is on the right side. And you can actually map the spatial distribution of this cilium here thanks to the chemograph that is at the bottom of the slide. So when the laser is off, you're not moving the cilium. And once you turn on the laser, you actually can move the cilium. And you can see that you can also control the frequency of bending that you're opposing to the cilium with the optical tweezer approach. So this is a highly controllable and tunable way to test mechanical sensation. So to combine gentle calcium imaging and fast imaging of ciliary dynamics with optical tweezers, we decided to build our own microscope that we decided to call CilioSpot for a ciliary selective plane illumination microscope with optical tweezers. And here, I'm not going to go into the details, but just for you to be aware, the blue lines represent the light sheet and the red light represent the optical tweezers. And this allows us to actually image the left-right organizer of a zebrafish in a very controllable and tunable manner. And in the field of view, we have all of those cilia of the left-right organizer, which is composed of about 70 cilia at this stage, and this allows us to only bend 1 cilium. So first, to test ciliary mechanical sensation, we needed to also know what were the endogenous dynamics of cilia at the stages where they're mostly immotile. So we decided to do differential interference contrast imaging to study the endogenous dynamics of cilia. And here you can actually see, thanks to those 2 chemographs, with the A prime representing a cilium on the left and A double prime with a cilium on the right, that the cilium on the left seemed to be subjected to a higher displacement. And we quantified those dynamics, and we saw that the mean displacement frequency was at about 1 Hertz for both sides, but that the cilia on the left side of the structure were subjected to a higher angle displacement. So we decided to reproduce those parameters using our optical tweezers. And we did that in a context where we first paralyze all cilia to not be biased by any type of endogenous ciliary motility. So here, cilia are not moving unless we come and oppose mechanical stimulation with the optical tweezer. And I'm going to show you whether, and those cilia are also expressing genetically encoded calcium indicator to follow those calcium dynamics, in addition to a red fluorescent protein that allows us to do ratiometric measurement. So here, let's see whether cilia respond to the mechanical stimulation when we bend them with the optical tweezers. Okay, so I'm really sorry. You should actually see that the cilium is moving and you have a huge rise of the green signal in the cilium. And here you will be able to actually appreciate this response thanks to this chemograph that shows the response. Oh, okay. I'm really sorry, my computer is actually really refusing to cooperate today. But here, just for you to know, the cilium is responding. So yes, it really seems like cilia in the left organizer are capable of responding to a mechanical stimulation that is opposed by the optical tweezer. And on the video on the right that is refusing to play, you're supposed to see a second response. So here, I think you could appreciate the multiple responses. And you have another one that is supposed to appear. So just to show you that when you keep a cilium and bend it multiple times, you will have multiple responses induced by the optical tweezers and the mechanical stimulation. So our recordings are not trivial data to analyze, because we're recording at a 7 Hertz frequency and we're recording for many minutes. So most of our recordings represent hundreds of frames. So to analyze those data, we decided to take advantage of machine learning to train a machine to actually extract these variation of fluorescence over time for us. So, but first you have to train the machine. So I've been working with Chris Nguyen at the Mass General Hospital to be able to train a software to extract those variation of fluorescence over time for us. And we designed a software that we called CiliaNet, which is a machine learning based tracking software of calcium dynamics in moving cilia. And here, just for you to see how it works, we give him an input, the video with the raw recording, and the output is this region of interest that is opposed on top of the cilium. And at the end, we end up with those traces that you see here. And here, these traces represent the cilium that we saw, the movie of the cilium that we saw last. And the orange arrow represents the start of the optical stimulation. And you can see here in the green trace that we have multiple responses in this cilium, highlighted by the red stars. And you also see that there is no increase in the rate trace, and that those increases are just in the GCAM signal. We also determined that we ended up with those responses after about 34 oscillation of the cilium after a displacement of about 2.4 micrometer and an angle displacement of 30 degrees. And we also remarkably noticed that when we bend cilia in the left-right organizer, in the left or in the right side of the structure, we actually end up with responses in both sides, meaning that regardless of the location in the left-right organizer, all cilia have the intrinsic ability to respond to mechanical stimulation, but the one that display those responses endogenously are the one that are subjected to the flow that is there. So we also observe that in addition to increases of calcium in the cilium, we notice that when we bend the cilium, we also observe increases of the signal in the surrounding cells, in the mesendodermal cell, meaning that the calcium rise that we observe in the cilium is actually transmitted in the rest of the mesendoderm. And we quantified it here with this graph, meaning that the cilium here is really the entry gate of the calcium signaling that leads to the proper lateralization. And as I mentioned earlier, told you that those endogenous calcium oscillation rely on the polycystin 2 channel. So I repeated those experiments with the optical tweezer in the absence of the polycystin 2 channel. And you can see here that when we don't have polycystin 2, cilia don't really respond to those optical stimulation, meaning that polycystin 2, strongly suggesting that polycystin 2 is the mechanosensor allowing cilia to respond to those optical stimulation. So now that we actually see that cilia can sense ciliary mechanosensation, we wondered, is ciliary mechanosensation instructive of left-right patterning? And to answer this question, we once again have the context where all the cilia are paralyzed, and we wonder whether one cilium would be enough to instruct proper left-right patterning. So here, the experimental setup is the following one. All the cilia are paralyzed, and we decided to bend only one cilium on the left side to determine whether this would be enough to rescue proper lateralization. And we decided to do that at the stage where we see those endogenous calcium oscillation, which is between the 1 and 4 somite stage, which covers a window of 1 hour. So we optically bend one cilium for 1 hour between the 1 and 4 somite stage, and wanted to know whether with this would be enough to rescue proper laterality. So here, in a context where cilia are not moving, as I told you, you're supposed to have complete randomization as shown in this bar. But when you come in a structure that have paralyzed cilia and bend only one cilium in the left-right organizer that is composed of about 70 of them, and when you come and bend one cilium for an hour on the left side, this is sufficient to rescue proper laterality. So you're forcing the heart to be sided on the left side by only bending one cilium in this structure. And knowing that we could actually do that by directing lateralization with one cilium on the left side, we wondered whether the opposite would be true by bending one cilium on the right side and see whether we could force lateralization on the right side. And it turns out that yes, you can. When you're actually going on the other side and bend one cilium, again, in a structure that is composed of about 70 of them, you can force lateralization on the other side, truly strongly suggesting and demonstrating that ciliary mechanosensation is instructive for proper left-right patterning. So I'm happy to report that if you want to know more about those results, we published those results a year ago. So please, if you need more detail, refer to the study, or you can also reach out. I'd be more than happy to discuss this. So to summarize, we demonstrated that oscillatory mechanical bending of left-right organizer cilia results in ciliary calcium influxes, that PKD2 is required for ciliary mechanical sensation, that LRO ciliary sensing is necessary, sufficient, and also instructive for proper left-right asymmetry. And I think we also show that optical tweezers are a powerful tool to probe the role of biomechanical forces during development. So all in all, what's the next step? So we actually want to understand what are, in addition to polycystin 2, what is the molecular machinery responsible for ciliary mechanical sensation? And we're currently trying to understand the role of PKD1 like one, which is another polycystin 2 channel, which is a binding partner of polycystin 2 in the left-right organizer. So we are trying to understand how PKD1 like one and PKD2 cooperate to allow ciliary mechanical sensation in the left-right organizer. We're also trying to understand how is ciliary mechanical sensation transduced into the cell body. And beyond left-right asymmetry, we're also interested in understanding whether cilia function as mechanosensors in other tissues and developmental processes. So with that, I'd like to thank all the people who have been involved in the study. And the first person I'd like to thank is Xiaolu Yuan, who is my current advisor, for trusting me in accomplishing this work. I'd like to also thank all the people that we collaborated with. I also like to thank the mentors that helped me during the past few years. And last but not least, I would like also to thank the SDB Ethel Browne Harvey Postdoctoral Seminar Series organizer for allowing me to share my work with you today. And once again, I apologize for the lag in movies, but yeah, I guess that if you want to see the movies, yeah, you have to check out the publication, or we can set up another Zoom soon. And with that, I'd be happy to take any question. Thank you.
Thank you, Lydia, for the wonderful talk. That was a really beautiful story, answering the long-standing and fundamental question in developmental biology. So we already have a question in the Q&A chat. So the first question is, so this is a very neat system. I was curious if you could modulate the bend of the cilia. I guess getting at is there a certain bend angle required that will restore asymmetry when all the cilia were paralyzed?
Yes, you could actually modulate the bending per se. So I didn't try. The only thing I kind of decipher was the number of bend that were needed to induce response, but I didn't really, and I also quantified the amount of displacement and angle of the bending. But yeah, it is actually doable to change the direction of the bend. It is with the optical tweezer. They are allowing us to do that.
Yeah, I also have a question. So
yeah,
so what kind of mechanical information is actually being sensed here? So like, is it amplitude or is it like frequency? Do you have any idea for that?
I think it's mainly the amplitude perhaps, and also the number of bending per se, because one push is not enough to induce a response. You really need to have multiple ones. And we think that the reason why it's the case is because this structure is subjected to a lot of noise, and that the cilium needs some type of thresholding system to be able to decipher what's noise and what's a true cue to be taken into account. Yeah.
Mm. Okay, and another question we have is, does the bending of a single cilia induce calcium signaling in the surrounding mesenchymal cells?
I'm sorry, say it again. I didn't understand this question.
Does the bending of a single cilia induce calcium signaling in the surrounding mesenchymal cells?
Yes. Yeah, that's why I tried to show with the video that the bending of one cilium allows also an increase of calcium signaling in the mesenchymal cells.
And another question from anonymous attendee is, awesome work. How do you think the activation of the mesenchymal cells is achieved? Do you think it is mediated by membrane tension or via cytoskeletal components?
I think it actually might be achieved via calcium-induced calcium release, and there are components in the basal body of the cilium that would allow the calcium to be released. So we don't really know per se what's going on at this level, how the calcium is transmitted, but there are a couple of candidates there, and inversin is one of them. that is actually at the base of the cilium. Because in inversing mutants, you have a loss of these, or you have a disrupted calcium activity.
Okay. So the next question is, have you looked at the time series of gene expression during your bending experiments, like lefty nodal?
We only looked at one, DEN5, and we actually also saw that we could direct the expression of DEN5. And so when we're bending on the left, DEN5 is actually right-sided, and when we're bending on the right, you'll have a left-sidedness of DEN5. So yes, we didn't look at nodal per se, but we looked slightly earlier than that.
Thank you. The next question is, many mutants seem to compromise flow, but do not affect laterality. Do you think your results suggest very little flow is required to initiate the cascade? Beautiful talk.
Thank you. I do think that, yeah, a little bit of flow is enough to initiate the cascade, because you see those calcium oscillation at the stage between 1 and 4, where when you have mostly immotile cilia, you have a little bit of motile one. So I actually believe that, yes, you don't need a lot of flow to initiate the cascade. And I think that the strong flow we see at the end of the left-right organizer development is actually there to ensure that proper laterality will take place. So I think that everything is initiated by little flow at first, yes. And that's something we're also trying to determine, how little and what's the amount of flow and what are the flow dynamics at this stage.
Thank you. Another question we have is, amazing. Will it be possible in the future to do multi-optical tweezers while recording calcium of the whole left, right?
Yes.
Over?
Yes. Yes, that's something we're working on. To not only have one trap, but multiple at once, yes.
Great. Yeah, so we have one comment for you, so take a look at that later. And so if you have no more questions, thank you, Lydia, again for. Oh, sorry. Oh, there are a question in chat. Yeah, so we have a question in the chat. So do you know if the laser of the optical tweezers generate heat on the cell?
So that's something we actually checked. And with the amount of laser power we use, we don't think that we're inducing non-specific responses, because we did assess for that. And we decided to go for the lowest amount of laser power that would allow us to move the cilium, but not generate any heat or non-specific like responses. Yes, that's something we assessed for and we controlled.
Right. Thank you very much, Lydia, for the wonderful talk.
Thank you, Yuki. Thank you.
Hello, everyone. Thanks, Yuki, and thank you, Lydia, for excellent talk. I have privilege to introduce our second speaker today, Dr. Granton Jindal. Granton received his PhD in the lab of Professor Stanislaw Schwartzman and Professor Rebecca Bedin from Princeton University. His thesis involved analyzing pathogenic MK variants in zebrafish. Granton is currently a postdoctoral researcher in the lab of Professor Emma Farley at University of California, and is investigating how mutations in non-coding areas of genome affects heart-specific gene expression using high-throughput assay in Sionna and human iPSC cardiomyocytes. Granton has been very productive and received multiple competitive fellowships and awards, including Sulman Award for Cardiovascular Research. He has published 11 papers so far and many more in pipeline, including one he will be talking today. Granton aspired to become an independent researcher and is currently looking for faculty position. The subject of his talk today is single base pair changes within heart enhancer dramatically increase binding affinity and disrupt heart development. With that, Granton, we are looking forward to your talk. Take it away.
Thank you, Rashmi, for the kind introduction, and thanks to the organizers for giving me the chance to present my work today. So I'm very interested in figuring out how sequence encodes function for enhancers in the context of embryonic development. And one of the enhancers I'll be talking about is active in the cardiopharyngeal mesoderm of the Siona embryo, shown here. So enhancers are regions of the genome that act as switches to turn on gene expression at the right time and place. So here you can see the red heart progenitor enhancer turn on its gene MESP in the heart progenitors. And the purple notochord enhancer switches on the gene brachyury in the developing notochord. The human genome with 22,000 genes has on the order of 1 million of these enhancers. And these precise patterns of gene expression encoded by enhancers allow a fertilized egg to develop into a complex multicellular organism with a head and a heart. However, we need to understand enhancers at the sequence level because small changes in enhancer sequence can have dramatic effects. So here we have a very famous limb enhancer, the ZRS enhancer, which drives a very precise expression pattern of sonic hedgehog in the developing limb bud, shown in purple here. And this is required to give you 5 fingers on your hands. Just a single base pair change from G to A leads to the mutated enhancer driving expression in a less precise manner, which you can see in this schematic, of sonic hedgehog expression. And this ultimately leads to extra fingers. So Siona is an excellent organism to study how developmental enhancers encode their function. They have fast development, so in 8 hours, they undergo gastrulation and form specialized structures. Additionally, reporter plasmids can be electroporated into fertilized eggs, and we can visually tell which cells express the construct because the embryos have a fixed lineage with a small number of cells. Siona embryos then undergo metamorphosis to reach their adult form of a filter feeder. And although they look very different from you and me, Siona are a part of the urochordates, the closest living relatives of vertebrates, and share many of the same developmental programs as vertebrates, and that give rise to many similar embryonic tissues, such as the notochord, neural tissues, and the heart. So in this talk, I will be focusing on how heart developmental enhancers encode their function. So the FOXF enhancer contains several ETS transcription factor binding sites shown in blue, which just indicate the presence of a GGAA or a GGAT motif in the DNA. Embryos electroporated with an enhancer reporter construct express M-cherry fluorescent protein in the cardiopharyngeal mesoderm, as shown by the red signal in the image. So we turned to protein binding microarray technology to look up binding affinities for each site to the ETS transcription factor. So previous work was able to measure the binding affinities to the ETS transcription factor by using protein binding microarray technology. The double-stranded DNA on a microarray is labeled with fluorescently labeled DUTP, so each spot you see here is a piece of DNA, and the signal from yellow being the brightest to blue being the lowest value is how much DNA is present in a spot on the microarray. The transcription factor binds to the double-stranded DNA and causes an Alexa 488 signal in places on the microarray, with yellow being the brightest and blue being the lowest value. So at this DNA spot, for example, there is bound ETS transcription factor. So this resulted in a data set with binding affinity scores for each transcription factor binding site, indicating the strength of interaction with the ETS transcription factor, with 1.0 being the highest value and 0.07 being the lowest value. Using this data set, we scored the binding affinity of ETS sites in the FOXF enhancer and found that their binding affinities ranged from 0.09 to 0.24, which is very low relative to the binding affinity of the consensus sequence of 1. So the wild type FOXF enhancer turns on in the cardiopharyngeal mesoderm cells. And the enhancer with the cores mutated does not drive any expression. So incredibly low affinity ET sites are necessary for expression, but then we wondered if individual ET sites were necessary for expression. So the wild type FOXF enhancer turns on in the cardiopharyngeal cells at a certain level, and the construct with no ET sites drives no expression. And the enhancer with the ETS1 mutated drives significantly lower expression than the wild type enhancer. And the rest of the sites significantly lower heart expression to varying degrees, except ETS6, which does not have an effect. Interestingly, mutating the ETS3 site had the largest impact, likely due to nearby ATTA and MESP sites in green and purple, which are activator sites necessary for enhancer function. So individual low affinity ET sites are necessary for heart expression, but we then wondered what would happen if we increased the binding affinity of the ET sites. So since deleting the ET6 does not have an effect, we remove it from the schematic. So there are 5 functional ET sites. So optimizing the binding affinity of these 5 sites leads to ectopic expression in cell types receiving RAS signaling, which is the signaling pathway that phosphorylates and activates the ETS transcription factor. And this has been seen with other studies, including ones published from our lab on the nervous system and notochord in Sionna. But this construct has 16 base pair changes, and the previous studies use multiple base pair changes as well. Since most changes in the genome are single base pair changes, we wondered what would happen if we increase the binding affinity of an individual ET site with a single base pair change. First, we determined the base pair changes that would modify affinity of the 5 ET sites and found that there were 4 mutations that had an affinity fold change of 3 or higher. So the largest fold change in ETS1 is a T to C single nucleotide variant, or SNV, that optimizes the site almost 8-fold. And we were very surprised to see a single base pair change that did this. ETS2 didn't have a base pair change that increases affinity significantly. The largest fold change in ETS3 is a T to G SNV that optimizes the site 3-fold. And the largest fold change in ETS4 is a T to C SNV that optimizes the site 3-fold again. And ETS5 didn't have a base pair change that increases affinity over higher than 3. So again, the wild type FOXF enhancer turns on in these cardiopharyngeal cells. The FOXF enhancer with a single nucleotide variant that optimizes the binding affinity from 0.12 to 0.97 drives expression in the cardiopharyngeal cells, the ATM cells, which are the 2 non-heart muscle cells, and the mesenchyme of the embryo. The FOXF enhancer with the ETS3 T to G SNV that optimizes the binding affinity from 0.11 to 0.34 drives expression in the cardiopharyngeal cells, the ATMs, the mesenchyme, and the brain. And the FOXF enhancer with the ETS4 T to C SNV that optimizes the binding affinity from 0.17 to 0.52 drives expression in the cardiopharyngeal cells and the ATM cells. So surprisingly, single nucleotide variants can optimize the binding affinity of ET sites and can cause ectopic expression in these two non-heart muscle cells, which will be important on the next slide. So how does this affect the heart? In embryos harboring the wild-type FOXF enhancer, a high concentration of activated ETs drives FOXF expression that causes the cardiopharyngeal cells to migrate to the ventral midline, shown here, and form the heart, as shown in this picture. In embryos harboring the affinity-optimizing SNV, FOXF is driven by both a high concentration and a low concentration of activated ETs that causes all four cells to migrate to the ventral midline to form the heart, and this is shown in this picture. So affinity optimizing SNVs cause cell migration defects. But to what degree? Specifically, embryos harboring the wild-type FOXF variant do not have any cell migration defects. About 10% of embryos harboring the ETS1 T2C variant have cell migration defects. About 10% of embryos harboring the ETS3 T2G variant have cell migration defects, and about 8% of embryos harboring the ETS4 T2C variant have cell migration defects. We initially expected the ETS1 T2C mutation to have the largest effect because of the 8-fold increase in binding affinity of the site. But then we remembered that it is actually farther away from the ATTA and MESP sites in green and purple, which are activator sites necessary for enhancer function. So we hypothesized that the optimizations to 0.34 and 0.52 are working in synergy with the nearby ATTA and MESP sites to boost their contributions to phenotype. This could be an example of enhancer grammar, which is the interplay of affinity with organization of transcription factor binding sites in the enhancer. In a few days, they develop into juvenile Sionna, and this is the wild-type single chamber heart beating, shown here. In juveniles harboring the affinity-optimizing SNV, we see a range of defects, including expanded hearts to a two-compartment heart phenotype, shown here, where there are two separate chambers beating within one pericardium. This is a striking phenotype for one example, and we wondered if we could find affinity-optimizing variants in other Sionna cardiopharyngeal enhancers. So we look at putative Sionna cardiopharyngeal enhancers defined by regions of the genome that are in open chromatin in cardiopharyngeal cells. Sionna cardiopharyngeal enhancers contain low affinity ET sites, as shown here, with an average of 0.12 binding affinity. 44% of these enhancers have a site that can be optimized to a binding affinity at least 3-fold or higher by a single base pair change. And the distribution of the resulting ETs binding affinities are shown here. How does this relate to humans? Cardiac enhancers within human fetal heart contain low affinity ET sites, as shown here, also with an average of 0.12 binding affinity. 61% of these enhancers have a site that can be optimized to a binding affinity at least 3-fold higher by a single base pair change. And the distribution of the resulting ETs affinities are shown here. So we find that a majority of human fetal heart enhancers are vulnerable to mutations that optimize binding affinity by a factor of 3 or more. We next validate one of these affinity optimizing mutations in a human fetal heart enhancer. So in this putative enhancer region in humans, we find a conserved enhancer that has been previously validated in mice. The wild-type GATA4 G9 enhancer drives expression in the mouse heart, as shown here. When 4 of these sites are mutated, as indicated by these X, the enhancer loses its ability to drive heart expression, as shown here. So we, for the first time, score the binding affinities of these ET sites and find that they are all low affinity from 0.09 to 0.12. So this shows that incredibly low affinity ET sites in the mouse GATA4 G9 enhancer are necessary for expression in the mouse heart as well. So it is known, another thing, it's known that excess GATA4 in mouse hearts can lead to cardiac hypertrophy. So we wondered if affinity optimizing mutations within the human enhancer could affect reporter activity in human-induced pluripotent stem cell cardiomyocytes. So in human cell lines, it is difficult to assess tissue specificity, but we can monitor levels. So here is the single base pair change that optimizes this site from 0.11 affinity to 0.73 affinity, a fold change of 7. We also tested a variant where we optimized 4 sites. And then we did a parallel reporter assay to characterize the gene expression levels driven by these enhancers. Both mutations yielded significant increases in enhancer activity, which means that affinity optimizing mutations lead to gain of function expression. So to conclude, we show that the FOXF enhancer needs incredibly low affinity binding sites. If we get rid of the sites, we lose expression. Also, affinity optimizing single base pair changes can optimize affinity of a site by a factor of 8, causing ectopic expression, causing cell migration defects, shown here, and causing heart defects. We propose that mutations like this could form the basis of evolution of a 2-chambered heart from the single-chambered heart in Sionna. And we find affinity optimizing SNVs cause gain of function expression in human cardiomyocytes. Altogether, variants that optimize the affinity of existing transcription factor binding sites could be prioritized to be the causal enhancer variants in disease. I would like to finish by thanking my mentor, Emma, and the rest of the Farley Lab, especially Alexis and Joe, who are co-second authors. I would also like to thank Aga and Kelly Frazier, who helped us perform the IPSC work, and Steve LePage, who collected the Sionna for us. Also, I would like to thank my sources of funding. And with that, I would be happy to take any questions. Feel free to reach out to me by email or Twitter as well. And also thanks to the SDB again for inviting me.
Thank you, Granton, for excellent talk. And now we are moving to Q&A session. I will start with very basic question I'm curious about. So, Granton, you mentioned several low affinity binding sites present in enhancer region. So I'm curious, do they interact to each other and work in a cooperative manner or additive manner?
Yeah, so there are many low affinity binding sites in the FOXF enhancer, and what's clear is that if you mutate one of them, you do see lower expression. But then if you this was work data I didn't show, but if you mutate a few, like 3 of them, if you mutate the first 3 sites, it completely turns off, it completely turns off function as well. So they are working together, and I don't go into in this work how this is affecting phase separation of the enhancers, but something like that could be happening as well.
Interesting. So I have one question in Q&A. Dave is asking, have you tested your GATA4 mutant construct in transient transgenic mouse to determine if it changes the expression pattern or cause heart defect?
Okay, this is a great question. We haven't yet, but this is an area of future research that I'm interested in pursuing. So so this is the GATA4 constructs of course are from the paper but by Brian Black's lab, and so we would, in order to test your question, we could do transient transgenic mice with the reporter with the enhancer driving GATA4. We could also knock in these mutations into the endogenous locus and try to see if that has a phenotype. So these are areas of future directions where the work can go. Absolutely.
Okay, so we have another question. Could affinity optimizing variance explain certain EQTLs?
Yeah, so this is another interesting question that is being actively worked on in the lab. It's not my project, but yeah, this could be the case. Yes, thank you.
Okay, I have one more question. So does this affinity of binding size changes over developmental time or tissue context?
Oh, could you repeat the question?
Does this affinity of binding size changes over developmental time or tissue contact, like the affinity of binding change over developmental?
I think I understand. Yeah, if I understand the question correctly, it is if enhancers that are on early in development, do they have different affinities maybe than enhancers that are on later on, specifically in a tissue type or something like that? Yeah, that could be the case. So there are enhancers that are on in very mature cell types that could have different affinity constraints than enhancers that are on pretty early in development, as to what I'm studying. But this is an area of future research that we could look into. Yes, thank you.
And I'll say the normal question in Q&A. So I can go with one more question. So does ETS site interact with other binding sites present in the enhancer?
Yes, thank you for that question. So there are previous work by Brad Davidson has found these ATTA sites that are also present in the FOXF enhancer and necessary for function. It's also known that there's a EBOX site when mutated away abolishes enhancer function as well. And so yeah, all of these sites sort of cooperate to cause the tissue specific expression, and there could be other sites as well that that we don't know of yet. Another way to kind of figure this out is to mutate is to basically mutate windows of the enhancer and see if we can find any more potential sites that are necessary for enhancer function as well.
Interesting. Thanks. So I think there's no more question in Q&A. If no more question, I will close. Thank you, Lydia and Granton, for your excellent talks. This seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, February 9, when Emilio Mendes Olivos from University of Calgary and Lauren Cote from Stanford University will present. Thank you all for coming.